Alrighty, well, hello everyone. Professor Kyle again, back with another lecture. Today we are going to be covering central nervous system. Now, full disclosure, 2 points. Number one, I'm recording this the day after Thanksgiving, so if I seem a bit more lethargic than normal, just blame the turkey and the pie. I'm having some jasmine tea here. This is my third cup of tea, so hopefully the caffeine will help me get through this relatively rapidly. Secondly, I previously recorded a solid portion of this lecture where I was essentially replicating what I did during lecture uh, in class, which meant more physio examples, more tangents, side stories, that sort of things, just to add a bit more color to the information that we're covering. That being said, though, I recognize at this point in the semester, this video being specifically for my students, you guys are just focused in on preparing for your last practicum exams. You want just the information you need to know for the purposes of the exam. So that being the case, I'm going to try and do this a bit more quickly than I otherwise normally would. And hopefully you guys are going to play this at like 1.5 to 2 times speed anyways, but hopefully that'll make this process a little bit more efficient. That being said, let's go ahead and hop on in. All right, so to start things off, we have this very terrifying slide right here. Okay, wait, give me my laser pointer. There we go. We have this very scary looking slide right here. This is a slide that essentially summarizes our cranial nerves. Now, as you should already know, we have 12 pairs of cranial nerves, pairs because we have 2 halves to our brain, 2 halves to our body, right? And so we have one for each half. These 12 cranial nerves are associated with a variety of our sensory and motor functions, basically for our cranial region. So everything from our hearing to our eyes to our face, muscles, that sort of stuff, our sense of taste and smell, as well as some eminences that go down to other portions of the body. We have our accessory nerve controlling muscles in our neck, and then we have our vagus nerve, which is a primary regulator of our parasympathetic nervous system, which is our fight and flight response, those sorts of things. We'll discuss that a bit later. But as you can imagine, this information is very important, so please dedicate it to memory. You're responsible for basically knowing the names of all of the cranial nerves, their associated Roman numeral, so like their number 1 through 12. As well as the basic function of the nerve. Nothing really more complicated than what is listed on this slide. So just mental note, basically dedicate this slide to memory. And we'll go over this silly little mnemonic at the end, but it's a very helpful memory tool. That being said, hopping on in, we'll start things off. We'll go top to bottom. So we have our cranial nerve 1, which is our olfactory nerve. This is just a fancy way of saying your sense of smell. In terms of the olfactory nerve, there's a couple structures that we should note. We have the olfactory tract right here, okay, as the name suggests, it's kind of a long tract. And then we have the olfactory bulb up here. The olfactory bulb, there are nerve projections emanating from the olfactory bulb that then would pass through the, if you remember all the way back from the beginning of the semester when we did cranial bones, we had the ethmoid bone, which had a specific feature known as the cribriform plate. So the cribriform plate has a little foramen kind of dotting its surface, right? It is through those small foramen that projections from the olfactory bulb pass through into your sinuses. This is your sense of smell. That was already too much information. Going to try and do this faster. Optic nerve. All right, optic nerve. Couple features we need to know. The projections of the individual optic nerves to your eyeballs would be coming out here. Those get cut off. That's fine. The other feature that we need to know is the optic chiasma. Okay, the optic chiasma. This is the point of convergence. So this X point, this is a point of convergence or a crossover in the cranial nerves because they get processed by opposite hemispheres of the brain. All right. Then we have a collection of 3 nerves that we should all kind of mentally associate together. Make a mental note of 3, 4, and 6. All of these nerves are associated with motor function of the eyes, okay? So ocular motor is responsible for controlling the majority of the muscles, and then trochlear and abducens are just controlling individual muscles respectively. So ocular motor, I'm not going to dive into this more because we're going to cover that in the next lecture after this when we do special senses and that sort of stuff. But these are the muscles associated with ocular motor. You are responsible for being able to recognize this on the test. I will relate a lot of what we discuss to the sheep dissection because we'll likely use the sheep dissection, or we are going to use the sheep dissection as a part of the practicum exam. Ocular motor is this guy right here, okay, is this little tab. If you remember from the dissection, you have the dura mater covering the entirety of the brain. Sorry, I'm going on a bit of a tangent, but this is exam related, so I'm justified. We have the dura mater that would normally be covering the entirety of the brain. When you had your sheep dissection, you had these large kind of projections coming off the sides, kind of each side of the inferior portion of the brain. Those would be the stalks of the trigeminal nerve, okay? And then under that and under the dura mater, you would be able to peer from the side, kind of looking from this side, and see the ocular motor. So the sort of geolocating facts that I would use to be able to identify the ocular motor, it is behind the optic, is basically in between the optic chiasma, or chiasm, and the trigeminal nerve, okay, the insertion of the trigeminal nerve. So under the dura mater, you see ocular motor. All right. Next, we have the trochlear nerve. Trochlear nerve is just controlling the superior oblique muscle. This is a very unique muscle within the eye. We'll discuss that more in our next lecture. Then we have trigeminal. We're going to skip that just briefly and jump to abducens. So again, abducens is cranial nerve 6, and that is controlling the lateral rectus muscle. Okay, so then that's why I put in this note here. Please relate that to number 3 and number 4, because those 3 cranial nerves together are controlling your eyes. We are predominantly, in terms of our perceptive bandwidth, we are predominantly a visual organism. The vast majority of our sensory input is associated with our vision compared to smell and hearing and all those other things. So they're very important. All right, circling back, we have trigeminal nerve. Trigeminal, as the name implies, has 3 different projections, hence the tri, meaning 3, in its name. Here we're just seeing the base insertion of it on the human brain illustration here. But this would then arc kind of forward and have 3 projections emanating off of it. Okay. Each one of those projections would run to a different portion of the head. So we would have the frontal, going tangent. We'd have the frontal portion, okay, frontal region of the brain. We would have another projection that is essentially along the maxillary region, okay, sensory and motor function. And then lastly, we would have a mandibular projection of it, okay. We already covered abducens, so we'll jump down to facial. Facial, as the name suggests, it's motor function of the face, okay? It is also one of 2 nerves associated with our sense of taste, so that would be the other thing. So 7, facial motor function, as well as part of your sense of taste. It's associated with a portion of the tongue. Then we have vestibulocochlear, okay? Vestibulocochlear over here, right? Complicated name, but it's actually very insightful with regards to the function of this nerve. So vestibule portion, okay, the vestibule is a part of your inner ear. This is associated with your sense of equilibrium, your sense of balance, your awareness of where up and down are, essentially, as well as momentum. Okay, so that would be the vestibular part of it. Then we have the cochlear part of the name. So cochlea, your cochlea are the other portion of your inner ear, and this is your sense of hearing, okay? So your sense of balance and your sense of hearing is emanating from cranial nerve 8, your vestibulocochlear nerve. Then we have glossopharyngeal, okay? Some muscles, salivary glands. This is the other one associated with your sense of taste. Leave it at that. Then we have vagus nerve, okay? The largest parasympathetic nerve, okay? It's down over here. What do I want to say about this? Essentially, this is your sense of fight and flight response. So imagine this as being a primary regulator. Your parasympathetic nervous system is a regulator of kind of your overall sense of homeostasis, what sort of state your body is in overall. As an example, after I had my lovely Thanksgiving dinner yesterday, actually lunch and dinner, it was great. Your body is in a restful state, all right? So my heart rate is low, my blood pressure is lower, it's under control, and blood flow is being shunted to my internal organs, specifically to my digestive system, to help transport those nutrients, right? Now, let's say, imagine I'm walking along, we are camping. The rest of my family is still camping currently. Camping, and all of a sudden I hear a rattling sound, and I look down, and I see a rattlesnake in the trail. All of a sudden, my fight and flight response would kick in, so my blood pressure would spike, it would increase in pressure. My heart rate would be elevated, and I'd be prepared to scream and run away, right? That's not actually what happens when I see rattlesnakes. I'm very mature and composed whenever I encounter small reptiles that want to kill me. I'm actually being serious. I'm a mountain biker, so I've run over many a rattlesnake in my day. Anyways, tangents, darn it. Then we have accessory. Accessory is very straightforward. Accessory is just controlling your neck muscles right here. Okay, very, very simple. And then we have hypoglossal. This is controlling your tongue, which is obviously very important for intonation of our words and stuff. Now, before I move on, a lot of information. Spent 10 minutes on a single slide. Don't worry, it's going to be faster moving forward. But given that's a lot of information, we have this very silly but useful mnemonic up here. All right, so I'm going to hop into a theater mode for a brief moment, get into character. I'll put on an accent too. Oh, 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 to touch and feel very good velvet. Ah, heaven, right? All right, so that little bit of theater out of the way, obviously it's very silly, okay? I'm very silly for doing that, but it is a very helpful mnemonic because it gives you the first letter of all of the cranial nerves in order, okay? So if you're trying to remember vestibulocochlear and which cranial nerve is that, because the question's asking me for what Roman numeral is associated with it, you can just run through that mnemonic, and then you'll eventually get to cranial 8. I'm not going to look at the slide. Yeah, cranial nerve 8. So please dedicate that to memory. It'll probably help you on the test. All right. Now, when it comes to the brain, there's a couple different ways we can try to define general regions of the brain. I promise I'm going to move faster. The simplest way that we're going to start off with is if you remember back to our cranial bones, all right, we have our frontal bone, temporal, parietal, occipital, all that sort of stuff. Well, if we essentially just map those cranial bones onto their associated regions of the brain, that is essentially how we're naming them according to this set of mnemonic rules. So we have our frontal lobe, just as we have our frontal bone. We have our parietal lobes, okay? We have our temporal lobes, very simple. The only one that's kind of a bit unique, kind of a bit different, is going to be the occipital, all right? So the occipital lobe, these naming conventions are applying specifically to the cerebrum of the brain, okay? So this sort of very wrinkly, spongy-looking structure up here. It's not applying to the cerebellum or to the brainstem. We'll talk more about those separately. So with the occipital lobe, it's a bit different because obviously the occipital bone would be covering this entire region right here. In this case, it's the occipital lobe is just, my video is really lagging. Wait for that to catch up. The occipital lobe is just referring to the portion of the brain, of the cerebrum, that is covered by the occipital lobe. All right. Now, is my video caught up? Yeah. If we zoom in on a little cross-sectional area right here, we would see something that looks rather like this. So a couple features we need to know. The gyrus right here, okay, this is the ridge itself of the folds, the wrinkly, raisiny folds of the brain. Then the divots formed in between those ridges, in between the gyri, okay, would be called a sulcus or sulci, okay? US is very common as a singular ending in Latin. I is very common as a plural ending in Latin. So gyrus, gyri, sulcus, sulci. Now, if we have a deeper sulcus, oftentimes delineating these lobes in their general structure, we refer to that as a fissure, okay? Then notice we have gray and white matter right here. We have gray matter as the more superficial layer. We have white matter as the deeper layer within the central nervous system, within the brain. It's going to be different when we get to the spinal cord. Why is that? Well, gray matter, if you remember, is associated with your cell bodies, okay? So this is where the nuclei of the cell, all of the cellular machinery, your mitochondria, that sort of jazz, lives in this region. This is the site of integration, meaning this is where information is being transferred and processed between different neural cells. This is where the synapses are, right? Okay, so this is where information is processed and the cells are making decisions based off of the information as it passes through the sequence, okay? White matter, on the other hand, white matter is simply your information highway, okay? This is transporting information between different neurons in different portions of the brain. Now, associated with that, since we have cell bodies and we have proteins and amino acids, we have basically darker staining material within the gray matter of the cell bodies. And then conversely, white matter is actually primarily a fatty structure because these are myelinated axons. So those myelin sheaths have a lot of lipids in them. So they end up having a whiter complexion. That's the reason for the difference in texture there. Or coloration, I should say. All right. Then, of course, we can just look at the general structure of the brain itself, and we can notice that there are some pretty significantly different regions of the brain. So we have the cerebrum, which we've noted previously. This is your conscious mind, okay? We're just going to leave it at that. Then we have the cerebellum, okay? This portion on the posterior part of your brain. Think of this as being associated with mapping out complex motor movements. So everything from maintaining tonal posture. You're probably sitting in a chair right now. You don't realize it, but a variety of your muscles are contracting or have a tonal contraction to them in order to maintain your posture. It's also associated with mapping out much more complex motor movements, like the first time you ever ride a bike, right? You have to think about it consciously, the pedals, the handlebars, your sense of balance, all that sort of stuff. Eventually, you don't have to think about that anymore. The reason you don't have to think about it anymore is because the cerebellum has basically figured out how to do it by itself. Your brain doesn't, your conscious mind, your cerebrum doesn't need to be involved in it anymore. Then we have the brain stem. Okay, so the brain stem actually projects up into the middle of the cerebrum. It's just deep relative to it. We'll talk more about that on a separate slide. Now, here we are zooming in, and we have a mid-sagittal cut. So it's the same perspective as what we're seeing right here, but we're essentially bifurcating the brain into two equal halves, and we're seeing the associated structures within it. We'll go ahead and go kind of top to bottom. We have the corpus callosum right here. It is this big, thick, white layer of white matter right here. This is the information highway between the two hemispheres of your brain. Okay, so this is how the two hemispheres of your brain are talking to each other predominantly. As you can imagine, if you were to remove this, which used to be done to treat epileptic seizures and schizophrenia and those sorts of conditions way back in the yonder years of the early 20th century, you had a lot of weird pathologies and psychological manifestations as a result of that. It's very important to maintain the continuity between the two hemispheres of the brain. Otherwise, you can start to get weird sorts of things like dual consciousness presentations and that sort of stuff. Tangents. Then under that, okay, it's not labeled here. I don't know why the label got cut off, but this open space right here, this is one of the lateral ventricles. Okay, we'll talk more about those in a little bit, but this is containing cerebrospinal fluid. All right. Again, we'll talk about it more later, but I just want to note that because this chamber, so the roof of this chamber is going to be the corpus callosum. The floor of this chamber is this layer right here called the fornix. Okay, the fornix is a part of a system called the limbic system. This is sort of base emotions. Think of like anger, happiness, hunger, sex drive, those sorts of fundamental emotions. It's considered to be a very basic, fundamental, or sometimes primitive, depending on your perspective on evolutionary biology, part of the brain. That's why it's sometimes called the reptile brain, but that's a misnomer. Just think of it as being a very fundamental part of your brain, okay? Other structures we should note. So this generally purpley blue region right here in the middle, this is a part of still the brain stem. So this projection that we saw down here, okay, goes up into the middle of the brain, the thalamus. This is the information consolidation center. Okay, this is all that sensory input is getting shunted up to here, and then the thalamus is going to decide where it needs to go. And it's also associated with like your sense of memory consolidation, that sort of jazz. Then I'm going to jump over here. We have the pituitary gland. The pituitary gland is a primary regulator of your endocrine system. So this is your hormone system, all right? We'll talk about this more, not this semester, but next semester. We'll talk about luteinizing hormone, follicle stimulating hormone, and all their associated processes with developing secondary sex characteristics, regulation of testosterone and estrogen production, that sort of jazz. So it's a very, very important little structure. We'll zoom in on it in just a moment. On the posterior portion of the brain, still kind of attached to the thalamic region, we have the epithalamus, and we have this little guy called the pineal gland. The pineal gland is associated with production of a chemical called melatonin. All right, so you may have taken that as a sleep aid before. When you take exogenous melatonin, you are essentially doing the job of the pineal gland. This is associated with your circadian rhythm. All right, so your sleeping and waking cycle, and pineal gland releases melatonin to help induce your sleep state, kind of bring your body down into a state of rest. And if you take melatonin, then you're essentially trying to induce that state. I mentioned in class, I can't take melatonin because when I do, I get weird dreams, like very, very, very wacky dreams. So I don't do it. I'll just stick to taking a Benadryl because I have allergies anyways. Alrighty, other things we need to know. We have the pons and medulla oblongata as a part of the brain stem. We have some of our cranial nerves emanating off of the pons and stuff. Think parasympathetic function. We'll just leave it at that. All right, and then here we can see a cross-sectional area of the cerebellum. I don't think I have a better slide of this, so we'll talk about it here. So again, mapping out complex motor movements. We have a feature that we need to note. So the white matter of the cerebellum kind of has these very rather beautiful emanations from it, or as it goes to the gray matter of the cells. That white matter and that shape is called the arbor vitae, all right, which means basically tree of life. It looks more like a fern to me, but it looks basically like a tree. So please note that structure. So the white matter of the cerebellum is known as the arbor vitae. All right. Anything else we need to note here? No. Well, we have a lot of slides to cover, and so some of these slides will be redundant. Here is a zoomed in perspective on the pituitary gland. Okay, so we have the anterior and posterior pituitary gland. The anterior pituitary is the one releasing those hormones I mentioned earlier, the luteinizing and follicle stimulating hormone. You don't need to know that for this test, but just know that it is an important part of your endocrine system, which is your hormone regulation system. All right, yeah, it's fine. All right, here's actually a cadaver model. So instead of an illustration, this is an actual human brain. So we can see a lot of those similar structures. Anything else we need to note here? No, basically covered this. Oh, I will note. So we have this. This is the lateral ventricle. Here it is labeled. Okay, so again, the roof of that is the corpus callosum. The floor of that is going to be the fornix. Then we have that lateral ventricle itself. Underneath the fornix, we have what is called the third ventricle. You just have one third ventricle. It's essentially on the midpoint of the brain. Again, it contains cerebrospinal fluid. There's actually a thin layer of specialized epithelial cell within these ventricles that is producing the cerebrospinal fluid, which is then kind of filtering and floating throughout the entirety of the outside of the central nervous system, including down the spinal column itself. That's about it. Here's an inferior view, noting a lot of the same structures we've seen before. So we have that optic chiasm. We have the mammillary body. Don't really worry too much about that. It's just a portion of the thalamus, medulla oblongata. Okay, so part of the brainstem, and then the pons up here. So we can see a lot of these cranial nerves. You're not responsible for identifying all these things, but we can see there's a lot of cranial nerves emanating off of that. All right, superior and inferior colliculus. We could, or colliculi in that case. We could dive into this more, but the simple version of it is these two little lobes on the posterior portion of the brainstem, and they are associated with reflexes of the visual and auditory variety. So your superior colliculus is associated with your visual reflexes. The inferior colliculus is associated with auditory reflexes. So imagine you're sitting in the library and you're studying, and then someone drops their phone or drops a book or something, and so there's a loud sound, right? And then you instantly kind of cue in and look at that sound. The inferior colliculus would be tying into that, okay, to make you react and essentially quickly allow you to determine whether or not the unexpected sound is a potential threat or not. Here's an illustration of the ventricles. I added some labels that weren't there previously during class just to be a bit helpful. So here we can see there are two lateral ventricles. There are one for each hemisphere of the brain, up through the cerebrums, each hemisphere of the cerebrum. And then we have that third ventricle that I noted previously, okay, third compartment. And then we have a fourth ventricle that is then running along the cerebral aqueduct, which is then going to run down across the spinal cord. All right, now we're focusing in on the spinal cord itself. Congratulations. As far as human anatomy goes for the brain, we've basically covered it. We could really get into this. I'm not going to get too aggressive with it. Let's just talk about some basic pathing of information, just to make this information a little bit more applicable. So we have these little bulbs right here, right? So these are referred to as the dorsal root ganglia. All right, so these would be spinal nerves that are coming off of the spinal cord. This would be the anterior surface. This would be the dorsal surface on the back part. So the way this works is all sensory input information from your body, okay, is going to come in along these cranial nerves, and then it is going to run through the dorsal root, okay? So sensory in, okay, is all going to run along the dorsal root, okay? All motor function, efferent neurons that are carrying motor signals down to elicit movement of some kind are going to be, can I use a different color? Yes, I can. Learning something new every day. All motor function, okay, is going to run along the ventral root, okay? So this would be, call this motor. Oh my Lord, this is terrible. You get the idea, right? So that's a very important classification. So sensory input information is going to run along the dorsal horn. These ganglia, okay, the ganglion, this is where the cell bodies of these nerves reside, and they would have a projection running kind of down out this way, and then another projection running into the spinal cord this way. Okay, so but the cell bodies reside in the ganglia, and then they would have their axons running out to these regions. All right, let me clear off erase all ink on slide. Cool. And then switch back to the laser pointer. It's just easier. Now, you just need to know some general anatomical structures. Luckily, same naming conventions apply to both the gray and the white matter, okay? Also notice, to start things off, we have a reversal of what we saw in the brain. In the brain, we had gray matter on the outside, we had white matter on the inside. Here we have white matter on the outside, gray matter on the inside. That's because the information needs to get into the spinal cord. So first things first, it's going to be white matter. Then it will get integrated and processed at these cell bodies, and then information, will be shunted off from there, whether it needs to continue to ascend, or a motor signal needs to get sent back down in a closed loop on the reflex. I'm not going to talk as much about reflexes as I did in class, but you have certain, essentially, like if you burn yourself, like I was making eggs this morning, if I was to burn myself in the process by touching the pan like an idiot because I didn't have enough caffeine, that nociceptor of the temperature and the pain and the crap this hurts would get sent into the spinal cord. And then one portion of that would then shunt the signal all the way back up to my brain, where eventually I would become conscious of it. But because it's a very painful signal and this is a problem and we need to fix it now, the spinal cord itself will actually send down a efferent motor signal back to my limb to recoil my hand before my brain's even conscious of what is happening. So that sort of integration and information processing can occur within the spinal cord itself, completely independently of your brain. Fun fact. All right, anyways, back to anatomy stuff. We have some simple naming conventions. We'll start with the gray matter. For the gray matter, we have these different projections that are referred to as horns. So this lower portion would be the ventral horn, and then the lateral portion would be the lateral horn, and the portion on the posterior side would be the dorsal horn, because remember, posterior and dorsal are referring to the same thing. Ventral anterior is referring to the same thing on the front side. So we have the dorsal, lateral, and ventral horns of the gray matter. On the white matter, we call them funiculus. I forget what that translates to. Just remember they're called funiculus, okay, instead. But same naming convention applies. So we have the dorsal funiculus, we have the lateral funiculus, we have the ventral funiculus. That in combination with your understanding of what the ganglia are, again, this is where the cell bodies are, and how we have sensory input along the dorsal root, we have efferent along the ventral root, should be good. Now, next we have the pia matter, or I'm sorry, the meninges, with the pia matter, arachnoid matter, and the dura mater, okay? These are important for the purposes of our dissection, because now we are going to shift gears from kind of running on general anatomy specifically applied to humans. Now we're going to be talking specifically about sheep dissection and that sort of stuff, because that is pertinent for our exam. Here's a quick slide before I talk too much about the meninges. The meninges are a outer layer coating that essentially surrounds your central nervous system. So here we can actually see the dura mater lining the cerebrum of the brain, and it would do so along the entirety of the brain, all the way down the spinal cord, etc. This is the most visible and the most tough layer, so that's what we notice the most. The arachnoid matter during the preservation process for our sheep brains actually ends up basically getting dissolved, so we don't really notice that. And then you can see a bit of the pia matter. It's very immediately superficial to the brain tissue itself, and it's a very thin little layer, but it is technically there. All right, so that's what we basically covered there with our meninges. And then now we're going to start to see a lot of things that we've already discussed before earlier in this lecture, but just specifically applied to the sheep brain. So we have the hemispheres right here, okay? We have the gyrus, which is going to be the ridges themselves. Then we have the sulcus, which is going to be the little divots in between. Cerebrum, again, your conscious mind, as opposed to the cerebellum, which is mapping out complex motor movements, and the brainstem, which is more like parasympathetic regulatory systems. We have the longitudinal fissure. Okay, longitudinal fissure is, as the name suggests, is a mid-sagittal line that is essentially bifurcating the two hemispheres. Juxtapose that to the transverse hemisphere, which would be running along this line right here. Okay, so this would be separating the transverse transverse fissure is separating the cerebrum from the cerebellum. Okay, so this line right here. In this instance, the cerebellum has actually been physically, my camera is catching up, physically pulled down to make the gap larger. Normally it'd be sitting up closer. All right, other things we need to note here. Talked about that, transverse fissure, spinal cord, we already know what that is. All right, superior and inferior colliculus, we can see the two of those. So if we pull down on the cerebellum, all right, we're looking at the posterior portion of the sheep brain. we can see those fissures right here. So this would be the superior, and then this little guy right, I should maybe pen this. So this is inferior. I'm not an artist. And then superior would be these guys right here. Does it make sense? Can I stop drawing? Okay, thank you. Alrighty. And then the pineal body, we can't really see that here, but we can see that on the next slide. So if we pull even more on the cerebrum, we can then see the bit of the pineal gland right here. Okay. And then this would be the superior colliculus, and it's not labeled, but these are the inferior right here. Cool. Cranial nerves, these are the ones you're responsible for being able to recognize on the practicum, because again, the practicum will utilize these same sheep brain models that you did in your dissections as part of the test. So we have cranial nerve 1, or olfactory nerve. So we have the bulb itself. We can't really see much of the tract. Sometimes you can see a bit of the tract kind of projecting backwards into the brain, but here we can see the bulb. Cool. We have the optic nerve, okay, cranial nerve 2. We have the optic chiasm, that point of convergence and crossover. Cool. Infundibulum of the pituitary gland. So the pituitary gland is actually not here. It's been removed kind of when probably that dura mater and that sort of stuff got pulled off. Don't worry too much about that. Here, then next we have the ocular motor nerve. Okay, so we need to know that. Cranial nerve 3. Again, it's a small projection. It's going to be under the dura mater. Yeah. Then we have trigeminal. This is just the base of the trigeminal nerve, but normally it would be a fairly large projection. This is not going to be very good artwork. But we would see a branching like this of the trigeminal nerve. Makes sense? Okay, cool. Brace all one side. And then we have the pons, okay? Upper portion of the brainstem, medulla oblongata, lower lump, kind of lower ridgeline of the brain stem. And then we have the spinal cord itself. Almost done. Here's, again, just some mid-sagittal cuts. Now, I caught an error, which I'm just going to apologize for. I don't know how, who, or where, or why it happened, but I will take responsibility for it. There was a mistake on the PowerPoint in class. This was labeled as pituitary. This should have been labeled as mammillary body. And the pituitary gland in this dissection is actually removed. So I added this picture over here. You can see it's this fairly large projection on the bottom of the inferior surface of the brain. So please note that in your study. On this picture, the pituitary gland is removed. So this would be the pituitary gland right here, projecting from the hypothalamus. Again, apologies for any confusion on the studying side of things. So we already talked about cerebral corpus callosum, all right, this upper layer information highway between the two hemispheres. We have the lateral ventricle, that is this open space right here. All right, then the floor of the lateral ventricle is going to be the fornix, this guy right here. All right, fornix, again, part of the limbic system, kind of basic emotions, that sort of jazz. The thalamus is basically the center of the brain, all right. Hypothalamus would be kind of the inferior and anterior portion of it. The head of the sheep is facing this way, okay. And then pituitary gland we just talked about. Pineal body or pineal gland, this guy right here, it's labeled on this one. Again, produces melatonin, regulating your circadian rhythm. Cerebellum we talked about, cool. Arbor vitae, that tree of life structure looking thing, cool. And then spinal cord, yes, we know what that is. All right, here is basically a frontal cut. So this is like cut like this, right? I'm Eastern European. I have a big nose. It's okay, I know. All right. So in this frontal cut, the basic reason I just put this in, this is actually taken from a dissection video that you have access to through your lab resources. But I just wanted to show you what these ventricles look like from the frontal perspective. So it kind of looks like an angry ghost in my mind. Anyways, these are the two lateral ventricles, and then this is the third ventricle. Again, there's only one third ventricle. There's two lateral ventricles. Anything else? Longitudinal fissures technically here. It's just kind of hard to notice, but again, that's separating the two hemispheres. Corpus callosum, the roof of the lateral ventricles. Fornix, floor of the lateral ventricles / the roof of the third ventricle, which we already noted. Cool. And then here's a quick slide with everything listed out. So in summary, please know your cranial nerves, their associated functions, and their numbers. know the general regions of the brain on the human model, and then definitely know them all for the sheep brain as well, with all these structures listed out on these last couple of slides. That is the most likely information to be tested on for the purposes of the exam. Congratulations, we are done with a relatively fast version of this lecture. Cheers.